the PHE885 is a anti-BCMA CAR-T cell product. This is an autologous product, so in some ways similar to the approved agents, ide-cel and cilta-cel, with a similar target. And obviously, the BCMA-targeted cell therapies are a really exciting area in multiple myeloma therapy, but there are sort of a number of challenges that are still, I think, related to these therapies. One is that they don't seem to last forever. Most patients do relapse after some period of time. And then the other is that there's an extended period of time for manufacturing. So for most of the clinical trials for ide-cel and cilta-cel, we were talking about 4 to 6 weeks from the time cells were collected before they could be returned to the patient and treated. And that can be very difficult for a lot of patients with aggressive disease. So PHE885 has a novel manufacturing process that really shortens the time frame between apheresis and completion of manufacturing. So the actual manufacturing itself takes under 48 hours instead of the sort of 10 days to 2 weeks that we've seen with cilta-cel and ide-cel. And so what happens is you actually infuse a much smaller number of cells, and the cells expand within the patient. And so the other thinking behind that is that by having the cells expand and grow within the patient, it's sort of a more natural environment and may promote a more stem-like phenotype that may also improve persistence of the CAR-T cells and maybe improve their activity over time. So this is a phase 1 study that we're presenting updated results on. And so we've treated patients at 4 different dose levels, and the dose, we're still basically continuing with the dose titration and an increasing dose of cells, starting at 2.5 million cells, then 5 million cells, 10 million cells, and 14 million cells so far. So as of now, we've treated over 20 patients with this product at the various dose levels. We've seen very promising kind of early evidence of activity and expansion of the cells. So we can see pretty significant expansion of the cells in all patients measured by flow cytometry or by PCR-based assay. And we've seen responses in basically all but one patient. And that one patient that didn't respond was treated at the lowest cell dose and actually was really progressing to plasma cell leukemia at the time of the cell infusion, which really highlights the need for these sort of rapidly manufactured CARs, and that a patient like that would have progressed to plasma cell leukemia, probably may not have been able to be treated at all if manufacturing had taken longer. So we were able to get the product to them, but unfortunately didn't have a good response. And so the study is ongoing, and we're very excited, about continuing to see the sort of longer term, results and to see how deep and long lasting the responses are.